A very good evening to all. Am I audible?
Yeah, you are audible. Please start.
Very good evening to all. On behalf of Growing Seed Organization, I am Dr. Subhradip Bhattacharjee. Heartily welcomes you to this last day of our session. And I think that the session was pretty good for our the students who have attended this program, as well as I think the trainees also had enjoyed this program. Very our first training session. So having said that, I think as we have discussed with our also teammates, is one of our first training program, and we are also hoping for future training program of such kind in maybe in same other topics, some other topics in or maybe in different topics, especially regarding analysis in other subject also. So being said that, that this particular session as it was in based on biological data analysis, and as I was gone through that whole classes and the list of the topics based on vast list of topics we have covered, that our trainees have learned so many things from the trainers, and I think in coming days it will greatly help the people who have attended this program. So coming to that data analysis point of thing, I can say that as in the first day I have told you that data is the new petro money. You can say previously the place which has been held by the petroleums, people were earning lots of money using petroleums. Now that being replaced by the data science, each and every aspect of science as well as anywhere is nowadays running by data. And not only that, if we consider in organized education systems, we can see that there is a lack of uh, proper methodologies as well as proper techniques which particularly deals with data. Nowadays these things are getting improved, but still now there is not that much structured curriculum to teach data science and data related things. So due to these things, this kind of workshop and seminars and all these things, training programs on ad hoc kind of things are really helpful, as I can understand from my own experience as well as from that I mean feedback from other people. So having said that, that aspect of this training program was on particularly on that biological aspect as well as chemical science aspect point of view. And as we have received so many of feedbacks from your side, it's been quite good, and we are really happy with that the outcome of this training session. So based on that outcome, we are hoping that in coming days also we'll organize such kind of training session in maybe in other aspects like in I mean in geoinformatics and all this thing in geological sciences, remote sensing thing, the data science on that aspects as well as this. These things are also coming in the market. So having said that, without wasting much time, I would like to welcome Dr. Rajat Nath, who is an research scientist from Assam University, to deliver his doubt clearing session, I think, and the last day of this program. Rajat, I heartily welcomes you to this session. Thank you.
Thank you, Subhradip, and good evening, everyone. So today my internet connection is little bit slow. So am I visible and my am I audible to everyone? Am I audible? Hello.
Yes, you are audible, Rajat. Please start.
Okay. So in my last sixth class, I have taught about mainly in computer-aided drug designing or CADD. So we have seen there is two types of drug discovery. One is target-based drug discovery and one is ligand-based drug discovery. For ligand-based drug discovery, we are having a ligand of interest and we are finding a target for a particular disease. Or so sorry, there is some network issue in my side. So what I'm saying that in my last sixth class, I have taught about the target-based drug designing and ligand-based drug designing. And we have seen in ligand-based drug designing, we have a ligand of interest and we have find out the targets for this ligand. So we have done the ADMET, that is the absorption, distribution, metabolism, and toxicity analysis. Then we have do the mass analysis, prediction of bioactivity of the ligand. Then we have gone for the drug likeness analysis. And for drug target-based drug designing, we have seen that we have the target of interest, so that we have found the ligand to inhibit.
So for target-based drug design, what I am saying that when we are having a target and which is available in the database, we can easily retrieve this target from the database. But if it's the target is not available in the database, then we have gone for the homology modeling. And we have seen how from the amino sequence we can predict a quaternary protein structure. Okay. And then we have seen how to see the quality of the structure using the Ramachandran plot. And again, we have seen that is the after the getting the target and ligand, we have gone for the molecular grouping analysis, and we have seen how to predict the IC50 value of the ligand using the QSR. So for this sixth class, we have taught almost everything for CADD. Just we have not touched about the gene network analysis to identify the target because it is a very it needs at least 1 week for this gene network analysis, and we have not seen the molecular dynamic simulation because it needs a huge server to run a molecular dynamic simulation. So apart from this, the 6 days class on I have covered at least 80% of the theory. So if you are having any doubt regarding this, you may ask.
We have no issues regarding this class.
Thank you, Rajat, for this uh, for actually for joining in this situation. I think there are some network issue in your area due to heavy rain and all those things. So I want to ask the participant if there is any question you can ask. So else we will conclude the session. So probably there is no question for you, I think. So thank you, Rajat, once again for joining, and request you to kindly, if possible, please be there for some times. So
Okay.
now I would like to request Dr. Saurabh Singla to kindly join the session. Thank you.
Thank you, Mohan. I think I'm audible, quite audible.
Yes, you are audible.
Uh, okay, so yeah, I was asked to comprehend the sessions that I took before. So without taking much time, I'll go through in like little bit from the PPT if you want. But if you, I would like to know if like if somebody has questions before that, we will take that. If no questions are there, means I will uh, in like 5 minutes I will put up the PPT. or we can reply in the messages, like what if what will be preferable. If anybody has any questions or anything like that. Anybody want to ask something? And one more thing, I said that I will upload the PPTs, but unfortunately I could not do that due to different reasons actually. I'll try to provide in the group, okay? So I think, okay, again, then I will get to some basic questions I will ask. If I don't know, I have not went through rest of the sessions, but if you have went through the sessions for bioinformatics, then definitely you might have came across the package that is bioconductor. Yes or no? Have you came across the bioconductor package? Guys? No one. You know what is bioconductor package in R? There is a package in R that is called bioconductor, and in this package, quite a lot of work regarding bioinformatics to be specific has been done, and a lot more is being like a lot more functionalities, a lot more of functions came up in last few years under the bioconductor package. That is also a repository of packages only. So lot of new types of analysis are available. Whatever the latest research is being conducted regarding the statistical analysis of this genomics data, genomics data, a lot come up in the bioconductor package. And last time I also discussed about uh, Learn R package. Learn R package is also one thing that last time I thought I will tell. Uh, so, if possible, let me put up the something from the Learn R itself. Will be better. Please. Okay. Okay, so let's go straight for the PPT. In few minutes, I will quickly finish up the things again. That would be, I think, better option. So in one of the lectures, we discussed about comparing the means. Uh, we discussed what is the meaning of statistically significant data, how sampling affects the user percentages, how if a person is taking a user sample, the difference between two samples could merely come from the uh, sampling, and due to not due to actual reason, but just because of some random causes, and to test whether the difference is coming due to a specific reason or it is due to the random causes, we need to test the differences statistically. So when you are testing only two groups, then t-test is sufficient to do. Here we have worked out with an example that is a gender weight. In this, weight of male and weight of female, that is only two groups are there. So that was being compared, and the data was arranged like this. These are 3 columns of the data. First, you can see 40 rows and 3 columns. First is the ID column. Second is the group, which is telling if the data belongs to male or female. See, it is not showing all 40 rows, just few rows from the top, and here are the means for male and means for female, standard deviation for female, standard deviation for male. Okay, so go ahead. This is a basic t-test that could be done in case of a um, comparison for 2 groups. Here, this is the comparison itself, and you got a quite highly significant result, which is telling you true difference in means between group F and group M is not equal to 0. Okay, that is telling you that there is actually some difference between the weight in the group. Okay, so that's how it is. That's how it works for the 2 means. However, when you have more than 2 groups, then you have to go for an ANOVA. Then when we have more than 2 groups, then we have 2 types of causes that are causing the variation. And the purpose of using ANOVA is basically to separate out the variation caused by assignable causes and non-assignable causes. And this is a user structure of ANOVA. You know, there are several types of ANOVA. One-way ANOVA, two-way ANOVA, three-way ANOVA, factorial ANOVA, repeated measures ANOVA. So it is based on the requirement you can run. Okay, so whatever is the requirement of a person, based on that, he or she can proceed with the analysis part. Okay. Then this is how I showed that how we ensure that within a block there is homogeneity. I also told what is a block. Block, concept of block, I think, okay. So blocking means to say a group of homogeneous units that is being constructed for conducting a design of experiment. And this is a uniformity trial data in which the field is separated in small grids, and the yield in milligram per hectare has been calculated using the formula and the field data. And based on that, a different color of dot has been put for that particular grid point, and based on that the field has been classified into the different homogeneous groups. Okay, so this is a uniformity trial data that has been conducted in 2021 in a study. Okay, this is specific. It has been taken to exemplify uh, it has been taken to give you an example of how a block is constructed. Okay, so these are the assumptions of ANOVA. I told you that the population should be normal, the effect should be additive, variances should be homogeneous. If these are not followed, then transformations needed to be done. And there's to conduct transformations, there are so many options available in the software itself. So no need to worry about that, how to do and all. So here we did one example data in using the Agricola package. Agricola package is a quite like rich package for so many types of analysis. Not only types of analysis, we can generate so many types of design using the Agricola package. Uh, Let me try to run. Okay, just give me a second. I'll show you how you can generate a design. This is how it has been analyzed using simple CRD. Okay. If here you will give one more plus factor replication, then it will turn into an RBD. Okay. This is if this value p value is less than 0.05, then it is significant. Here it is less than 0.05, but not less than 0.01. So it is significant with single star. Okay, this is the code. And here you can see the pairwise, what you can say, pair post hoc analysis has been conducted. What is the post hoc analysis telling you? That the group T1 and T4 are almost at par. However, oh wait. Okay, let's go straight forward with the RStudio. Let me show you how a design can be generated. Okay, like firstly we can go with the library Agricola. So once, see, if I have already installed Agricola, if you do not have installed the library before, you have to run install.packages Agricola. So I have already installed, so I need not to install. I will cancel this. So I will just load by using library Agricola. This code you need to run only once. Okay. After that, see, if I want to generate a design which is a CRD. Okay, CRD design. So generating a CRD. I think I should increase the font size. Huh? Hope so it is better visible. So let's define a vector which is having treatments. So let's say we have 7 treatments. Okay, let's say we have 7 treatments. Okay, and let's say we have varied number of replications. Okay, so the replications are like 3, 3, 1, 2, 3, 4, 3. Okay, so you can see, let me show you how it look like. So this is how it looks like. Okay. So treatment 1 is having 3 applications, treatment 2 is having 3 applications, treatment 3 is having 1 application, 4 is having 2 applications, 5 is having 3, 6 is having 4, 7 is having 3 applications. See, this is only allowed in CRD, that is varied number of applications. Okay. So let's design a CRD which is having TRT is equal to TRP and R is equal to, that means replications is equal to repli. Let's supply the vectors and let's store it into the CRD layout. So see what is there in the CRD layout. There is, okay. So here you can see the software has provided you with. Firstly, it is telling you what is the design. It is CRD. These are your treatments. These are the number of applications for each treatment. And here it has allocated each treatment to a different plot number. Okay. So remember, CRD is a single block design. Okay. So you need not to give any field layout kind of thing, but assume that it is an RBD. Okay. Assume that it is an RBD. So let's put a layout for RBD. Okay. RBD layout design RBD. Let's say I have same 7 treatments, but the number of replication let's change. Okay. Okay, here it is RCBD. RCBD treatment is equals to TRT, and number of applications now we cannot change. Okay, this is fixed in this type of design. Also, there are so many other options. Let's not discuss. Let's not get into that deep today. So here it is an RBD design. Layout is ready. Let's see what is there inside. Okay. So let's see what is there. Oh, see, here is a sketch. This is what we call a sketch. Let's have a better look on the sketch. Okay. Let me put sketch and on and let's transform it. Okay. So here you can see you have got perfectly there are 7 treatments in each block. If this is on the vertical side, there are 7 treatments. Okay, and on the horizontal side there are 4 applications. So you have got an automatically randomized layout of a RBD experiment, in which on this side you have blocks and on this side you have replications. Like in one block there are all the treatments: 1, 2, 3, 4, 5, 6, 7. Again it is been randomized: 1, 2, 3, 4, 5, 6, 7. Again 1, 2, 3, 4, 5, 6, 6 and 7. Same for the 4th block. So likewise you can generate so many designs. Okay, here I will show you what all the options are available in case of this. There is option to go for design of an alpha lattice design, incomplete block design. Okay, this is CRD. This is a cyclic design you can go for. Then you can go for a Greco-Latin. Then you can go for a lattice. You can go for a Latin square design. You can also provide a design matrix of a custom nature. You can also design a split plot, strip plot design. Jordan square design also you can design using this package. So these are all built-in functions. Okay, that means you need not to randomize manually for the each and every step. That is the advantage of this specific package, and using this package for the designing of the experiment. Similar thing is that I used here in the PPT. I will show you. Let me. Okay, here I will show you. I have used the same package. See, here I have used Agricola only. This is Agricola to conduct the post hoc analysis. This is LSD.test. LSD test you can run once you have a linear model object. This is a linear model object, LM. So you just need to provide LSD or linear model object and what you want to compare. Here I wanted to compare treatments. So once you give this input, that is you can conduct the post hoc analysis as well as the ANOVA. ANOVA is for running ANOVA, you don't need Agricola package. ANOVA is built-in function, but to conduct this post hoc analysis, definitely Agricola package is quite helpful. Then in another class, I shared about the second part of the analysis, but like this is more or less the part. If you have more queries, something like that, you can ask anything. Even if you have any other doubts in statistics, in if not even not if related to this PPT, you can ask. It is from my side, the session is open. That any doubts you have in specific analysis or your particular your own analysis, you can ask. No problem.
Thank you, Dr. Saurabh. Now I am requesting Dr. Sanghamitra Raut for the talk.
Thank you. Good evening, all. So, I'm just going to give a little bit of brief what I have covered in my session. So, what two topics I have covered. It's about principal component analysis and cluster analysis. So, if any one of you are having any doubt, you can directly ask now, so that I can try to solve your questions. You can also write in your chat box. It's all about this PPT and code sharing. Just after the session, I will try to send it in the group itself, or we'll share it so that you can directly use those codes for your analysis. And in future also, if you have any data set and you face any problems, you can directly contact us. We can help you in doing that particular data set analysis. Yes, if you are having any doubt, please ask. Fine. I think there is no question. So I just brief up a little bit about my presentation of principal component analysis. So I have talked about why it is being used, then its applications, you can say, then PCA methods for factor analysis, what are the steps involved in the PCA. Then we have discussed a little bit about a real-time example by taking some real-time examples and their data set, how to do the analysis manually as well as by using the R softwares. Apart from that, we have also discussed about the visualization of the graphs of PCA, which includes your chord plots, a biplot, factor loading, variable plots. So I have discussed all those in a brief, like how to interpret it, because that is also a very important part when you are writing your research paper. So principal component analysis, as I already told, that the main advantages is to reducing the number of variables in your data set, and also to remove any kind of external factors like any noise ones which you want to omit. So you can use the PCA for it. Apart from that, you can also use it for data visualization and improving your algorithm performance by focusing only on some relevant traits. Okay, so these are some of the advantages, and here what we are seeing is your in visualization, what all plots we are seeing is like your scree plot, in that you will get a bar type of graphs, which actually shows your which principal components, which all principal components are showing the greater variance. And then comes your biplot, which includes both your variables as well as if you take some genotypes or any traits, both of them can be placed together. Like we have taken one example for PISA data collection, right? We were having a set of PISA data set, and we have done a biplot which PISA will be better, which brand pizza will be better, and they are also having their nutrients. So we were comparing nutrients and brands in our example. So which pizza brand will be better, having the better nutrients. So likewise, such type of interpretation, if you want to do, you can do it easily with your biplots. After that, we have discussed a little bit about this PC applications, like it is being used for image compression, noise filtering, feature extraction, or data visualization, right? So this is all about your PCA, and we have also run the code for PCA data analysis. For that, the basic thing is you have to install the libraries which are required for it, like your facto extra, facto minor, metan library, or if you want any color graphs, for that you have to install your library of R color breed. From there you can see a palette and color palette, and you can put that in your graphs for better visualization. Okay, so these are some libraries which are being required for your data analysis, and obviously I will provide you the codes which you can run directly and see how your data set is being visualized. After that, we have discussed about cluster analysis. So cluster analysis is it can be done in various methods are there to do that. But what I have shown you is the Mahabalano-based D-square analysis, in which you have to firstly develop a D-square matrix, that is the distance matrix, from where you can see how much distance is between the two genotypes. So actually, why we are doing this is it's just the simple, like if you are having a large number of genotype and you want to see the genetically diverse parents in that particular data set. So for that, you can go for this cluster analysis, Mahabalano-based D-square analysis. From there, you can get your diverse genetic parents for your hybridization program. So in that, we have also discussed about dendrograms. So on the basis of this dendrogram, by looking at the dendrogram, you can easily sort out which cluster is having how many genotypes. So by looking at that, we can say easily how the genotypes are diverse from each other. If they are falling in the genotypes are falling in the same clusters, that means they are less divergent. And if they are falling in the different clusters, that means we can say that they are more distant from each other. And selecting such type of genotypes from different clusters, we can go for our hybridization program in our agriculture. Similarly, if your data set is of different background, you can also do the same dendrograms. You can prepare the same type of dendrogram and do the clustering and give your interpretation. Apart from that, for preparing this dendrogram, you can do it in a bar graph way, or bar dendrogram, or in a circular dendrogram also you can represent by using the codes. So all these codes are actually available in Google also. You can see there which pattern of dendrogram you want for your data set. So this was the basic things which is being required for a good presentation of your research paper. And by doing this, it will be more easy to interpret your data also. So if anyone still have any doubt, you can ask me.
हेलो मैम।
Yeah, sure.
मैम एक्चुअली ये होता है ना कि बहुत बार फैक्टोरियल काइंड ऑफ डाटा होता है, तो उसमें ग्राफिकल रिप्रेजेंटेशन कैसे करते हैं, ये सारी चीजें नहीं समझ में आती है। अभी जैसे मेरा बायोकेमिस्ट्री बैकग्राउंड है, तो जैसे
कौन सा
इसके मीन्स डाटा के लिए कौन सा ग्राफिकल रिप्रेजेंटेशन? कुछ बार जब वेरिएबल बहुत ज्यादा होते हैं तो हम पीसीए वगैरह यूज करते हैं, बट फैक्टोरियल काइंड ऑफ डाटा जिसमें बहुत सारी ट्रीटमेंट होती है, फिर सब ट्रीटमेंट होती है, तो
इन दैट
उसमें
केस
ग्राफिकल रिप्रेजेंटेशन या फिर मीन्स पीसीए या ये चीजें कैसे करते हैं ना, ये समझ में नहीं आता है। तो मैम मीन्स अगर मैंने आपको डाटा का काइंड ऑफ स्ट्रक्चर भेज दिया तो आप मुझे मीन्स बाद में सपोर्ट कर सकते हैं कि इसमें किस टाइप का आप प्रेजेंट
हाँ,
मीन्स?
देखो अगर आपका कितने फैक्टर्स हैं, लाइक 3 4 फैक्टर, 3 फैक्टर है, 2 फैक्टर है, एज पर दैट तुम अपना अनोवा एक बना लो पहले, उसके बाद फिर उसके बाद यू कैन ऑल्सो गिव अ बार ग्राफ टाइप ऑफ ग्राफ भी बनता है, आप उसको भी बना सकते हैं, या फिर बॉक्स प्लॉट टाइप ग्राफिकल प्रेजेंटेशन भी आप दे सकते हैं। सो देर इज नो इशू इन दैट. So if you have any problem, if you show me your data, I could help you in doing at least for one trade. I can show you.
यस मैम, यस मैम। ये अगर मैंने आपको फिर मेल में कर दूंगी
हाँ,
डेटा का,
sure,
फिर
no,
यस
no
मैम,
problem. You can
ओके
share
मैम।
your data set. And as per that, you can also properly mention like हर trades का तुम्हें अलग अलग graphical presentation चाहिए, या फिर combinedly चाहिए, या फिर दो factors को मिला के तुम्हें graph बनाना है. Those things you mention in your in the mail. I will help you.
यस मैम, यस मैम, थैंक यू मैम।
Oh, sure. Thank you. Yes, anyone else are having any kind of doubt? O- Okay, then thank you. I think if you face any problem, you can directly mail me also. I will help you in your data set, any kind of analysis which I could able to do. Okay, thank you so much.
Thank you, ma'am, for this valuable insight and doubt clearing session. Now, I would like to request Dr. Rupshanathan Mandal to address the queries from the student side in the last session. Welcome, sir.
Good evening, all of you. If participants have any queries, please ask me. So, I think the participants have no doubts regarding the software, whatever I have explained, or rather than it may be not applicable for all the participants. That's why they have no question. So if sometimes later or even after, when you are practice that software, if you are facing some problem regarding that software, whatever you have learned in this training, okay, you can email me or WhatsApp me. So thank you, all of you. Have a great day or great night. Okay, good night.
Thank you, sir, for this session. Before we conclude this session, we have a few things to discuss as well as declare that regarding the certificates, we will issue that offline certificates only, and to collect those informations which are required to send, provide the certificate there as per that NSDC requirement. So we will prepare a Google sheet and we will share with all the participants. You must carefully fill that Google sheet and then send back it to me. We will do it within few days, and hopefully we will be able to dispatch the certificates within 1 week. And more things about our organization is that apart from this training institute, as you are a little bit aware of, I think you are aware about our organizations, that we are a non-profit social welfare organization, and we are very much into that research and development, especially in agricultural sciences. So if anybody wants to do any kind of collaboration with us regarding research and development, they are totally welcome to our organization. And apart from that, we also publish one monthly magazine, e-magazine, named Agri-Alice, which has been published since 2019. We are publishing articles. So you are also welcome to submit your popular articles to our magazine. And as I have said that in research and development, we are trying to establish collaboration throughout India in different fields, particularly in agriculture and relevant subjects. So if anybody wants any kind of information and collaboration and anything like that, we are happy to be a part of having a collaboration with you. In this regard, we are always open. And most importantly, that about the certificate, you should submit the details, whatever is been asked to you. That sheet will be shortly shared with you. And all the participants are also requested to submit the feedback, not about the single class, but about the whole program, so that we can improve our program for that future programs also. In that regard, we will share the documents. And if anybody has any doubt, you can ask now to anybody. Our tutors are already present. So if you have anyone have any kind of suggestion or anything, you can ask that to now. Otherwise, I will move to Dr. Manik Bhattacharjee, convener, Indian Science Congress Organization, Dharmanagar Chapter, for the vote of thanks. But before that, if anybody has any kind of anything to discuss and any feedbacks to give, you are free to discuss it. Thank you.
हेलो सर, मुझे एक बात कहनी थी कि जैसे ये अभी 21 डेज का वर्कशॉप था, इसमें अगर हर एक डे का हमें रिकॉर्डिंग या फिर पीपीटी मिलता तो वो अच्छा हो जाता, क्योंकि सेकेंड डे हम उसका प्रैक्टिस करते फिर वो थोड़ा समझ में आ जाता है एकदम। आप मीन्स सारी जो रिकॉर्डिंग्स है या फिर पीपीटी है, ये सारा कोर्स के बाद अगर मिलता है तो उसका उतना यूज नहीं हो पाता। ये थोड़ा सा मैंने एक दो बार मैसेज भी डाले थे ग्रुप में कि हमें अगर रिकॉर्डिंग्स उसी दिन की मिल जाती तो फिर वो प्रैक्टिस के लिए और फिर चीजें और समझ में अच्छे से आ जाती थी। ये थोड़ा सा मुझे मीन्स मेरे लिए थोड़ा सा टफ था ये सब, तो इसलिए आप अगर रिकॉर्डिंग और सारी पीपीटी अभी दे देते तो वो सही रहेगा सर।
हाँ, ओके मैम, हम कोशिश करेंगे एज सुन एज पॉसिबल रिकॉर्डिंग प्रोवाइड करने के लिए। हम इसमें काम कर रहे हैं अभी फिलहाल, हम शेयर कर देंगे मैम आपके साथ में ये जो भी क्लास हुआ है इसके रिकॉर्डिंग्स।
ओके सर।
एनीबॉडी एल्स? Okay, so I think nobody has any kind of doubt or any discussion left. So now I would like to invite Dr. Manik Bhattacharjee, convener, Indian Science Congress Organization, Dharmanagar Chapter, for vote of thanks. Dr. Manik Bhattacharjee, sir, you are welcome to this conversation.
Respected chairpersons of this session, respected participants, respected scholars, respected resource persons, at first, on behalf of ISKA Dharmanagar Chapter, I want to convey my thanks to all of you. And particularly, I want to convey my thanks to Growing Seed for their contribution and for conducting very important workshop for the young research scholar particularly. ISKA Dharmanagar chapter, we have started our journey from 2017, and all of you know there are 28 chapters in India under Indian Science Congress Association. Among them, we are ISKA Dharmanagar chapter, and particularly we, the ISKA Dharmanagar chapter, conduct different types of scientific awareness program all over Tripura, national seminar and workshop, and this program is a part of our annual activities. And we actually organize different types of program with the help of Growing Seed. And Growing Seed organizes this particular workshop with collaboration with ISKA Dharmanagar chapter. So I again want to convey my thanks to particularly Sohanab, Poonam Bhattacharjee and all other members of Growing Seed. And in a remote place of Dharmanagar, they are trying to the research scholar of different parts of India. And again, want to convey my thanks to all resource persons, all participants, all scholars, and all persons who are directly or indirectly involved with this particular workshop. So thanks to all.
So I think it was that last message from our side, that once again we will be asking you to provide your information regarding the certificates, and as we can see that there is no more doubt or anything to discuss, we are hoping that whatever that lecture has been provided by our lecturers and trainers, it will be helpful to the students. And with that, I am closing this session. Thank you, everyone. Thank you for your presence.